All right, good morning, everybody. Thank you very much for joining our program here today. Laura is urging me to start on time and stay on time. So, once again, thank you very much for coming out today, and I hope you learned something about what WARF and ICTR is, planning on, and hopefully this will just be the beginning of our conversation. So, in just a moment, I'll be introducing, Dr. Allan Brasier, Director of ICTR, and he's going to talk about his goals in drug discovery and development. I'm going to then take over and lay out WARF's vision for WARF Therapeutics. We're going to take a short break at 10:30 after we have a little Q&A between Allan, myself, and you all. We're going to convene at around 11 o'clock, and then I'm going to be joined on stage by Doctors Jennifer Golden, Doug McNeel, and Luigi Puglielli. Each of these individuals are participating in academic drug discovery research. They're all at different phases, and so I think it's going to be a very interesting discussion. So before I turn the podium over to Allan, I just want to kind of help set the stage. So today you're going to hear about the complexities, the risk, the high attrition rates of the drug discovery process. You're going to hear about when and why drugs fail in the clinic and the lessons learned from pharma and how they've responded to these lessons in order to become more efficient, to be able to get more drugs across the finish line into patients and helping to modulate debilitating diseases. But more importantly, Allan and I are going to describe our respective organizations, how we've aligned our thinking to collaborate with each other in order to collaborate with each and every one of you more effectively, in order to move your biological discoveries across the valley of death and into the clinic and hopefully into patients. The take-home message is this. ICTR is going to be able to help contribute to the early and the late translational aspects of drug discovery. And WARF Therapeutics is going to help bridge that gap between these early biological discoveries and what the pharma industry is looking for in terms of a licensable clinical asset. And so hopefully by the end of today you'll understand that we'll have end-to-end support for the entire drug discovery process, from the novel target discoveries, the target validation, as well as the preclinical drug discovery aspects. I'd now like to introduce Allan. Allan received his MD degree from the University of California in San Francisco, completed his internal residency at the Brigham and Women's Hospital in Boston, and subsequently completed a clinical research fellowship in adult endocrinology and metabolism at Mass General and the Howard Hughes Medical Institute. In 1991, he joined the University of Texas Medical Branch as a physician investigator in the Department of Medicine. He advanced to full professor in 1997 with tenure. And for the past 12 years, Allan has served as the director of the Seeley Center for Molecular Medicine. And over the past 10 years, he was principal investigator of the UTMB, Clinical Translational Sciences Award, and founding director of the Institute for Translational Science. Allan is internationally recognized for his original and high-impact research, has been elected to the American Society for Clinical Investigation, and was an established investigator of the American Heart Association. He served on numerous NIH study panels and published over 240 manuscripts and books that have been independently cited over 11,000 times by his peers. Allan serves on NIH peer review, as well as many other prestigious review panels. And in addition to his service for 6 years on the experimental virology study section. Allan's research has focused on the mechanisms of innate inflammation and its role in advancing pulmonary and cardiovascular disease. His work has been pioneering in applying advanced bioinformatics to integrate information from high-throughput gene and protein expression studies to generate novel mechanistic insights into innate inflammation. This past year, he was recruited to the UW-Madison to lead the Institute for Clinical and Translational Research as Executive Director and Senior Associate Dean for Clinical and Translational Research. ICTR is launching a number of new initiatives in team science, mentoring in systems biology, and promoting the applications of medicinal chemistry, which will, he'll be speaking about this morning. So without further ado, Allan.
Thank you, John, for that very warm introduction, and it's really great to see such a fabulous turnout for our program this morning. So I want to talk a little bit about some of the things that are going on within ICTR and some additional goals that we wanted to establish with this drug discovery program. One of the key outcomes I hope that we'll be able to accomplish is to help to establish a community of practice that's interested in medicinal discovery and medicinal chemistry and drug discovery. And my talk today, I'm going to talk a little bit about how ICTR sees medicinal chemistry as an essential component of its portfolio in being able to accomplish its translational research mission. To inform some of our panel discussions, I'm going to talk a bit about some of the trends in academics and industry with regards to medicinal chemistry and drug discovery. And we'll highlight some of the substantial resources that we have here at U of W to help to advance medicinal chemistry programs, including the alliance and partnership with WARF. And finally, we'll talk a little bit about some of the challenges with regards to medicinal chemistry. So the Institute for Clinical and Translational Research, as many of you know, is a partnership. It's housed administratively within the Health Science Learning Center at the School of Medicine and Public Health, but it's a partnership between 5 different U of W schools and colleges, including the School of Medicine and Public Health, the School of Pharmacy, the School of Nursing, the School of Veterinary Medicine, the College of Engineering, and we have a very close partnership with the Marshfield Clinic, which allows us to have substantial and wide outreach across the state of Wisconsin. So our mission is to discover, translate, and disseminate innovations in human health and health care. And in order to accomplish this, we're funded by several large grants, one of which is this Clinical and Translational Science Award, or CTSA, which John had indicated before. We also receive substantial funding from the Wisconsin Partnership Foundation, which helps us to realize the Wisconsin idea. So just to use terms clearly, we see translational research as being able to advance a drug or a device or a behavioral intervention across the entire research spectrum, from preclinical work, which would be viewed as being the T1 phase of translational research, into humans, ultimately translating into patients, into practice, and then finally being adopted by the community, where it's actually used in practice of healthcare. So we view the translational research spectrum is from the bench to the bedside to the curbside. Being able to bring translational research from basic science discovery into first applications in humans is a first step, but is not our end goal. Our end goal is actually to improve health outcomes, and that's really where our wins are. So I think you'll notice as we progress along this talk, we'll bring in the chevron diagram of the pathways for drug development and drug discovery, and you'll notice that there are substantial similarities between our translational research spectrum and also the drug discovery and development pipeline. So in this way, the end goals of ICTR with regards to focusing on improving outcomes in health involve some component of commercialization and entrepreneurship. And we see this as one of the aspects that a partnership with WARF will be quite valuable for both of our programs in order to be able to achieve these goals. So why is translation research such a focus? And I think that we have a lot of evidence that we don't do translational research very effectively or efficiently. So in the drug discovery and development arena, we know that the translational or the pharmaceutical development is very inefficient. Over 95% of the drugs will fail before they actually become used in people. and that over half of the drugs fail in phase 2 clinical studies for lack of efficacy. In addition, the costs are extraordinarily high, which makes this very slow and cumbersome and expensive. So part of the challenge of the CTSA and the Victor is faster and more efficient. And so we are working on the process of translation, which is to improve this activities. Faster, better, cheaper is our mantra. And so we were looking a lot at the aspects of translational research. So segueing over into some of the current trends in academia with regards to drug development and discovery, there have been some substantial changes in how academia is seen in the drug development arena. And the factors that have influenced this include the successful completion of the decades-long NIH Molecular Libraries Program, which I'll talk a little bit about. Changes in R&D investment, and particularly with regards to partnership with academia in terms of growth of translational research centers. The CTSA programs are one site for improving medicinal chemistry and its application, but there are also other translational research initiatives, including SPARC, the University of Pennsylvania Institute for Translational Medicine, and the QB3 at UCSF. So these activities have really shifted what academia's role has been from being primarily a site for where we identify targets and disease-relevant targets that can be subjects of medicinal chemistry, to also having substantial small molecule expertise, including enhancements of screening approaches and innovating new probes and approaches that can inform subsequent therapeutics development. So I'm going to talk a little bit about the outcomes of the NIH Molecular Libraries Program, because this has really changed a lot of the field by which we approach medicinal chemistry. So this was a program that was funded for about 10 years out of the Roadmap Initiative. A very nice summary of the program was published by Stuart Schreiber and his group in Cell. And these high-throughput screening programs contributed substantially to the expertise in academic medical centers. So a number of probes were developed which can be used for exploring different types of biology. The Molecular Libraries Program developed these fluorescence polarization high throughput assays, which I think are commonly used here at UW for looking at medicinal chemistry probes that may interfere with protein-protein interactions. They were instrumental in developing multiplex small molecule screens, as well as cell-based assays, which help to identify targets that have, or drugs that have some substantial toxicity early in the screening so that they can be dropped out of subsequent development. And then also identified that the scope of druggable targets was substantially larger than what was initially thought. So much of this is a diagram of the different types of targets that have been realized as a result of the Molecular Libraries Program. The G protein coupled receptors historically have been the easiest targets and the major focus of most industry and pharmaceutical development up to about 20 years ago. But as a result of the Molecular Libraries Program, new targets were demonstrated to be feasible, including targets that had to do with translational regulation, DNA binding and epigenetic regulators, tyrosine kinases and non-tyrosine kinases, regulators of small molecules and metabolites. So this expanded substantially the feasibility for being able to probe biological processes and has changed the scope of academics. In addition, pharmacy has also undergone a bit of trends in evolution in their perception, and John Talk will describe this in much more detail. One of which is this very well recognized productivity crisis. that 1 in 10 drugs entering the clinic are approved despite increased investment in the R&D process. So the investment is not resulting in any increase in the number of approvable drugs. And a lot of the reasons behind this include increasing regulatory complexity with regards to the clinical trials environment. This is an area where ICTR is working on. The costs of clinical trials and internal processes favoring industrialization with these high throughput screening facilities, which haven't been informed by deep knowledge of what the target actually does in a biological setting. In addition, pharmaceutical companies are investing in regional science hubs. So we know a lot about the Tres Cantos, the Pfizer Center for Therapeutic Intervention, and Sage Bionetworks with the idea of trying to produce more meaningful collaborations with the academic environment. And as John will talk a little bit about, the trend in industry now is that there's a greater emphasis on target validation. And that is, we need to have a deep understanding of the disease and the disease targets, which is an area where academia can really shine and provides a meaningful contribution to the drug discovery pipeline. So why would we be interested in medicinal chemistry for biological targets? And I think there are a couple of areas, not only necessarily associated with drug development, but others which can be used. We can use medicinal probes to perturb functions of interesting molecules in complex systems so we can get a greater biological insight. Also, some of the medicinal chemistries can allow us to image activity of the target in vivo or examine its activity in a biological or pathological state. And finally, some medicinal chemistry compounds can actually be lead compounds which will advance to therapeutics. So there are several different pathways for development that can be considered within the academic environment. If an investigator is looking at a target which he or she may think is important in a disease, there are some paths could be a purely academic path, which is focusing on creating generalizable knowledges and publishing and developing grants. So small molecule chemistry can help to advance this understanding and to leverage this academic research program to be able to come up with new insights and to be able to leverage and substantially advance their program. But there's also opportunities here for industrial partnerships, and these have a different motivator in terms of protecting intellectual property and thinking about clinical applications, but it's a different pathway, and it's one that an investigative team should consider early in the process of development before they get too far along and lose the opportunity to protect their intellectual property. So the drug development pipeline is this chevron which sort of gives you the impression that it's a very linear process, which is probably not the case. But I think that also the important part is that this is very similar to the translational research phases that the CTSA program is interested in. From very basic preclinical target discovery to validation, to lead ID and optimization, preclinical studies, so these would be the first in human type studies that we would talk about as the T2 or T3 phases of research, going into phase 1 through 3 studies in humans, and then ultimately developing regulatory approval. And there are a number of UW resources that I'll briefly mention here that can help investigators along various different stages of this drug development pipeline. So I'll point out a couple of these. These slides I think will make available to the attendees of this workshop in case you want to follow up with these. I just want to highlight that there are compilations of cores and resources at the OVCGRE at this link. But we also have a number of very specific cores and resource support for being able to do GMP and small molecule chemistry, including the Weissman biomanufacturing facility that's run by Carl Ross. So this is a GMP facility that will allow us to grow and develop biologic that would be acceptable for applications in human trials, including recombinant proteins and cell therapeutics. The Wisconsin Center for Nanosystems, or WISC-Nano, focuses on nanomedicine, and Xianpo Hong and Glenn Kwan, Lingjun Li, and Sandro Mikosi are the leaders of this nanobiomedicine facility. And the Z Pharmaceutical Experimental Station, which is expert in drug delivery and stabilization evaluation, and Ed Elder runs that program as well. There are also a number of support for small molecule screening and pharmacology that has been developed by the Cancer Center. The small molecule screening facility allows screening up to 400,000 compounds, as well as some computational expertise for looking at drug target interactions using structure-based designs. The Cancer Pharmacology Lab, Medicinal Chemistry Center, and these are all activities that are available locally for UW investigators. Through our association with the CTSA, through the National Center for Advancing Translational Sciences, so this is a newly formed NIH institute that supports the CTSA programs, has substantial interest in developing molecular libraries and advancing chemical compounds. They have a division of preclinical innovation, which provides support for research process. All of these are competitive and peer-reviewed, but provide expertise to help selected projects that have translational and health outcomes advance. In addition, the NCATS program supports the TREND, or the Therapeutics for Rare and Neglected Diseases. So these are US consortium that study these rare diseases and are very interested in looking at efficacy studies in humans that have rare diseases, and developing and showing proof of principle for these types of medications. There's an assay development and screening technology. They have a drug repurposing program called the New Therapeutics Uses Program, and they also have core technologies which allow for development of automation, compound management, analytical chemistry, and informatics. So there are a lot of resources that will help to advance U of W discoveries go along this pathway to commercialization and or drug development. I've mapped some of these entities along this drug discovery and development pipeline, and you can see that there's a substantial amount of resources here that will help us to get past this valley of death that John talked about, from the target discovery to the early proof of efficacy in humans. And so these are available, and for individuals who want to take this on by themselves can help to do this. But there are other approaches to develop partnerships, and CTSA program has seen that developing a close partnership with the industry is really quite valuable because the pharmaceutical companies can bring substantial insight and resources and technology to a translational problem. So if we take advantage of our expertise, we can come up with biologically relevant targets, and then we can use collaborations to help develop probes, drugs, or imaging reagents to advance our mechanistic studies. But also these partnerships can lead to a viable strategy for drug development, and the Human Therapeutics Initiative be one way of approaching this. So I think that the perception and the value of the academic medical center in terms of the drug development pipeline has been cemented by this study in Nature Reviews of the AstraZeneca experience in failures of drug development. And what they found was that the majority of drugs fail in phase 2 studies. Over 50% of the drugs fail in phase 2 studies because the biological target has not been sufficiently understood or studied in a relevant system. So the drug fails because they don't understand the targets or the biomarkers of its effect. So I'm posting that the academic research environment can play a unique role in helping to reduce these failures by increasing our understanding of the drug and its target, its role in disease, and to help to reduce these failures within the clinical efficacy realm. And a further deep dive into these drugs that failed the phase 2 clinical efficacy studies have shown that most of these are really because the target linkage to disease is not established, and that is because either the target has not been understood or it's been studied in animal models that are not relevant to a human disease. And so these are significant challenges with regards to academic efficacy studies to make sure that we understand this process and we're studying these in a rigorous and reproducible way and in an animal model that's relevant to disease. So John's going to talk a little bit about how industry is now thinking about selecting drugs that are appropriate for drug development. And this is the AstraZeneca 5 Rs. That is the right target, is the right tissue, the right safety, the right patients, and the right commercial potential. And this is the area that I think that academics can really contribute by being able to focus on the quality of the target and its role in the disease, which will reduce our efficacy failures. So having said that, there's still some challenges with regards to the academic industry relationships, and we might be able to talk about this in our panel discussion after our talks. These may be due to differences in motivations. As you know, the pressure to publish within the academic center sometimes interferes with the ability to develop intellectual property protection. So this has to be managed in a way that is appropriate for these partnerships and recognized up front. In addition, there are differences in experimental analysis. So the view of industry is that over 70% of our studies that are coming out of academic laboratories can't be reproduced. And so this is a real challenge for academics to improve the rigor and reproducibility of our studies in such a way that they can be reproduced and advanced. So I think in the area where we're going to need more efficient models for technology transfer and managing conflicts of interest, and this is some areas in which ICTR is working on. So I want to leave with a couple of opportunities for you. We are working, and over the next couple of months, we'll be announcing an ICTR strategic partnership in precision medicine. So we're working with the Human Proteomics Program to develop collaborations with translational research teams to apply multi-omics profiling. And one of our endpoints is to help to identify biomarkers of therapeutic response. And this is one of the areas that has been identified as a failure for further drug development. And we're also thinking about trying to come up with some programs in target validation, trying to expand how we think about and identify validated targets so that these can be commercially viable and developed into usable drugs. So I want to make sure that I thank Rob Hagan in the Office of Therapeutics Discovery and Development and within ICTR, and Laura Heisler, as Director of Programming in WARF, for their work in organizing this program, and we hope that it'll be very successful. So we'll segue on over to John's talk, and so I'd like to introduce John. Thanks. So John began in his academic training as a synthetic organic chemistry at the University of Minnesota Duluth, where he obtained his bachelor's degree in biochemistry. He joined the research group at John Still at the Michigan State University, where he focused on the development of a synthetic organometallic methodology. He moved to Madison as an NIH and a Merck postdoctoral fellow, so he's returning to U of W as a Badger. And he worked as a postdoctoral fellow in Stephen Burke's lab, where he worked on the total synthesis of erythrinolyte B, which is the A glycone of the antibiotic erythromycin. He began his industrial medicinal chemistry career at the Merck Research Laboratory, spending the first 10 years at the Rahway before moving to Boston to help build the new research site in the Boston Longwood area, where he rose to the ranks of director. He then moved to San Diego to join the Celgene Labs, taking on new scientific responsibilities, including building and leading the high-throughput medicinal chemistry, high-throughput purification, and analytical chemistry groups, in addition to having portfolio responsibilities. He helped lead the team to the development of the new drug modality called Protax, and his final stop before joining WARF was at Regulus Therapeutics, where he was, which is one of the leading companies in microRNA drugs, where he was the vice president of chemistry and a member of the executive team. During his 25 years, he is an author or inventor on some 50 publications and patents. In addition, he's led teams or contributed to the discovery and development of at least 8 clinical candidates that span the therapeutic areas of oncology, obesity, urinary incontinence, chemotherapy, induced nausea and vomiting, hematology, and autoimmune disease. So he's currently the head of WARF Therapeutics, and he'll tell you about his vision and the organization and how he plans to collaborate with principal investigators here at University of Wisconsin-Madison. So, John.
Thank you very much, Allan. All right, I'd like to, take a, sort of a brief rewind and go back to sort of, the early 2000s, when I was, in the New Jersey site in Rahway, working for Merck. At the time, Merck was envisioning building new research laboratories in the Boston area. Of course, that's not the newsworthy story here. Merck was a global pharmaceutical company, and at the time, they had international sites in Montreal, London, Rome, and Tokyo, in addition to 3 domestic sites in New Jersey, Pennsylvania, and San Diego. However, those sites were built strategically for regulatory and patient access. And so the Boston site was going to be the first time that they ever built a site from a strategic research perspective. It was chosen actually to be in the Longwood area to center itself and position itself between Harvard Medical School, Beth Israel, Brigham and Young's, as well as the Dana-Farber. And the idea there was to build intricate connections to those universities, to build those relationships, to interact scientifically, and hopefully forge collaborations for doing drug discovery, collaborative research. Sounds very similar to my current role, actually. And in fact, it was that experience in Boston that actually, gave me the experience on working with academics and to leverage those key biological discoveries and show that, in fact, those collaborations with industrial medicinal chemistry processes could be a viable way to move those biological discoveries across that valley of death and into the clinic. And so later on today, at the end, I'm going to highlight that a particular story that really influenced me accepting this particular role, and that was when a professor named Gary Gilliland, who was the key discoverer of this activating mutation in the JAK2 gene, and demonstrated that this was was prevalent in about 95% of patients with polycythemia vera. And he knocked on our door and asked me, John, are you interested in, take a look at this, new science, take a look at this target validation. Would you be interested in a collaboration to move this program forward? And that, collaboration, ultimately led to a best-in-class, dual JAK1/2 inhibitor for myeloproliferative disorders. And so I'd like to, from a high-level perspective, I've kind of dedicated my life to the discovery and development of key therapeutics for some of the most challenging and debilitating diseases. Really excited about trying to discover and improve, discover new therapeutics and provide relief to patients that are suffering from some of these challenging and debilitating diseases. But on most days, I really enjoy the passion of science. I especially love the iterative process of medicinal chemistry, finding an issue, coming up with a hypothesis, testing that hypothesis. My problem is trying to turn that process off at the end of the day or on the weekend. As my wife can attest to, I have a sort of a hard time moving into sort of the human race on the weekend. And so to solve that, I took up to sort of running, and I thought this was going to be a way to sort of relax, allow my body to rest, turn my brain off for a little bit. But I'm sure, as Laura in the front row here can attest to, that is in fact true. But unfortunately, what in fact happened was, as you're sitting, running through the mountains, going up a trail, you're getting into the rhythm of the run, and all of a sudden, those issues that you're trying to run away from, all of a sudden they start popping into your head, and this relaxed mind, sort of leads to a way to work through that problem. And so 10 miles in, you solve that problem, what am I going to accomplish in the next 10 or 20 miles? And so ultimately, I sort of found myself with a scientific passion, but with a sort of a running problem. And so it turns out this affliction is rather prevalent in the Boston-Cambridge area. So these are all my Merck running friends participating in a 200-mile overnight race. A few other Merck fellows that participated here actually north of Milwaukee in a 50-mile trail race. And so that's just a little bit of sort of what makes me tick. Moving on to sort of WARF Therapeutics, the three pillars of WARF Therapeutics, as we see them, are to invest, develop, and partner. And each of these play prominent roles in defining our overall mission, which is to partner with UW principal investigators interested in translational research, to invest not just financially, but intellectually, invest our time, invest our experience, to help discover novel molecules that modulate these validated targets that Allan was talking about, to improve human disease, and ultimately to partner with biocentric companies for development and commercialization. The WARF Therapeutics team will be a small group of drug discovery experts, drug hunters, if you will, that were trained in industry drug discovery facilities and have a proven track record of navigating the preclinical drug discovery space, moving programs from target validation through screening activities, lead optimization, and hopefully culminating with high-quality drug candidates for clinical development. If we do all of that correctly, that will help support sort of the overall goals and aspirations to continue this legacy that UW has on innovative, transformative therapeutic agents such as Zemplar and warfarin, to bring these medicines forward to treat patients with unmet medical needs. If we do that, the revenues will follow, and then those will flow back into the university system. Just like every good program begins with an idea, a hypothesis, every good collaboration is just going to start with an initial conversation. So I want to really lower the activation barrier for coming to sit down and chat with us. No paperwork, no pre-work. Pick up the phone, give me a call, send me an email. Let's just sit down and talk about science. At the end of that first meeting, we'll see where it goes from there. But so if you find yourself doing drug-related research, whether it's target identification, deconvoluting biological pathways, validating targets, or perhaps you have an interesting cell line and you've done a phenotypic screen, for example. You don't know the target, but you have an agent that nicely modulates that particular cell line and gives you the right phenotypic response. These are all great opportunities. And so I'm reminded, for example, of the Revlimid story from Celgene. As you might for those of you that might know that story, Revlimid, began from the drug Thalidomide, which, was, approved in Europe in the 1950s, but led to, severe birth defects. Eventually, somebody discovered that that particular drug, really made, multiple myeloma cells go away. They advanced it through, preclinical studies. They advanced it into the clinic, and in 2006, it was approved in the US. At that point, the mechanism of that drug was not known. It wasn't actually discovered until 2012 that it was binding to a protein called cereblon, which then recruited the E3 ligase and led to the digestion and phagocytosis of multiple myeloma cells. So whether you're doing targeted therapies or phenotypic screens, these are all good opportunities to work with WARF Therapeutics. But this isn't going to be for everybody. As Allan said, there's a number of different motivations on campus, and everybody might be motivated by something different. But if our goals are mutually aligned and we decide to take this journey together, I'm really excited to take that journey with you. So, the WARF Therapeutics difference. We hope that by building drug discovery and optimization capabilities, adjacent to your laboratories here at UW, we'll be in a unique position to identify the best novel target opportunities and initiate programs, early on. My team will be, implementing industry standard processes that encompass all phases of the preclinical drug discovery space, which will include stage gate criteria to enable disciplined decision making and ultimately help increase the probability of technical success. And so this is going to be based on the 5R philosophy that Allan alluded to from AstraZeneca, and there'll be more to follow later on in the talk. My team will be familiar with the final compound profile and data packages that pharma executives expect to see when licensing in either academic or biotech assets. And along with our experienced scientific advisory team that'll be comprised of pharma executives, we'll also have key opinion leaders on our advisory team that are leaders in strategic areas and have a demonstrated track record of success. And of course, to make this initiative a reality and to really give it teeth and improve our probability of success, WARF has made a substantial financial commitment to this effort. And so over the next 45 or so minutes, I'm going to step through my vision of WARF Therapeutics, how we'll bridge the gap between academic research and the biopharma industry. I'll describe an industry-tested portfolio management framework put forth by AZ based on their 5-dimensional philosophy. And finally, I'll describe the program submission process in detail, including the program submission form based on these 5 Rs. And by means of illustration, I'm going to describe do a retrospective study of the JAK2 program for polycythemia vera to show, what data was led us to believe that this is a well-validated target. What were the in vivo animal models that gave us confidence to move, to start a drug discovery program and ultimately move that program, into clinical, practice. And so, similar to what Allan showed earlier, the drug discovery process can be broken down into 3 distinct activities: target discovery, preclinical drug discovery, and ultimately drug development. These can be further broken down into target identification and validation, compound screening, hit to lead activities, hit to lead ID activities, lead optimization, development candidate nomination, as part of a preclinical safety and efficacy study. It'll then move into IND tox and ultimately into the clinic and FDA approval. Of course, this journey is complex. It can take 9 to 15 years from the inception to FDA approval and takes on average around $1.5 to $2.5 billion. Typically, PIs on campus here, reach out to WARF at the time when they're nearly ready to, disclose publicly and possibly even do a patent application. And so I just want to urge people that that might work for a lot of the activities here on campus, but WARF Therapeutics is going to require some subtle differences to that effect. And that's really because of the unique characteristics of doing drug discovery. For example, if you came to WARF Therapeutics at this point, you've discovered and validated a novel target. This is not something that we can patent. The patent office has made it clear that you can't patent nature at this point. And so, you might then think, well, let's do a high throughput screen. Say you identify a hit out of that screen. Chances are, if it came from a commercially available library, that's not a novel hit. And the best thing that you could get out of that would be a method of use patent, and that's certainly not the strongest type of patent that one can get. From a pharmaceutical perspective, and I know this from experience, if somebody reports a paper with a tool compound, for example, trained medicinal chemists can take that molecule, and I'll show you in our JAK2 story later on today. That is sufficient information to actually design novel chemical matter, and could actually induce more competition. And so ideally, we want to try to publish our patent applications as late as possible. From the time that you file, you get 20 years of patent protection. So the longer that you can delay that, the better off the protection will be for that particular asset. And so the overall proposed sequence of events from a high-level perspective is, as I said, pick up the phone, call me, let's have a pre-disclosure meeting, no paperwork. As this evolves and you proceed through the target validation process, let's have a disclosure at that point. But it's really just to disclose that novel target, that therapeutic hypothesis, and possibly submit that to WARF Therapeutics for drug discovery activities. But the idea would not be then to write a patent application at that point, but let's start the drug discovery operation. Let's start investing on taking that novel validated target through screening activities, identify novel hits, turn those into novel, patentable, licensable hits, and possibly even advance them to lead optimization. And ultimately, let's go ahead and file that patent application as late as possible. So over the course of the last 20, 25 years or so, pharma has undergone a number of different strategic directions. There was a time when big pharma sort of operated in an independent model where they performed all three of these distinct activities by themselves. They built very large biological facilities, enhanced their screening capabilities, increased their library content, and built large medicinal chemistry operations. While I was at Merck, I think we topped out at around 1,200 medicinal chemists across our 7 different research facilities. So huge operations. It turns out that this didn't help with the attrition rates. These infrastructures were very challenging to maintain and often led to inefficiency, stranded resources, and really helped to drive up the R&D costs. So, as with most innovative biological discoveries, happen outside of pharma, pharma finally realized that they're going to need to leverage external partners to share in that investment, to share that risk in order to survive. So how does academia fit into these models? It's well established that academia has been much stronger in the areas of basic research than big pharma. In fact, in the paper shown below, the authors here show that sort of public is in blue, industry, pharma is in green. And what you can see by this slide here is that over the course of the last 40 years, more than half of the over half of, 50% of the most 200 influential drugs over the last 40 years had their starts in academia. And I think that's pretty impressive. That's a pretty strong message. Now, as you went through the discovery phases and the development phases, the role of academia was diminished, and this is where industry really picked up. They have always traditionally been strong in drug discovery and medicinal chemistry as well as clinical development, and so this really makes sense. Sort of the issues here, however, is there's kind of a little bit of a paradox here. Most of these drugs are getting their start in academia, but they're not necessarily sharing in the financial reward. And so how can we change that? The real issues, as I mentioned, is at this stage of the game, you can't patent that target. Pharma has made it very clear that they're not willing to license such an early asset. There's a lot of attrition, as Allan said. 95% of molecules at this stage fail by the time they get to the end. And of course, once that publication hits the public domain, pharma can start to work on that target and apply that to their own screening decks and move things rather rapidly. And so what pharma is really requiring here is a de-risked asset to go along with that validated target. And so the collaborative model between pharma has been really successful over the past decade. Companies like Pfizer and Celgene have heavily invested in biotech companies to access their novel targets and platforms. These companies continue to invest, de-risk those assets, and ultimately advance them to the interface between drug discovery and the clinic. As Allan mentioned, by the time they get here, still 90% of those compounds are going to fail. But those odds are much better than the 2 to 4% of drugs at the early target space. So, what can academia do to become more attractive to pharma? How can academia resemble the pharma development model? How can academia bridge this gap, survive the valley of death, or what I affectionately refer to as the zone of chaos? And I think the solution is what WARF Therapeutics is presenting, and similar to what other research organizations in academia have proposed, and that is build drug discovery capabilities of an industry-centric nature close to the academic centers where these discoveries are taking place. And so WARF Therapeutics will invest in the identification of licensable chemical assets that pharma will value. And in this model, there are a number of potential assets. We don't necessarily need to bring things to IND tox or phase 1 clinical trials, for example. I think there's a sweet spot from early leads, late leads, and development candidates that pharma will be interested in. As somebody that worked in pharma and looked at packages for licensing, I think there's a lot of opportunity here. So if you think about this as risk and investment, it's going to take a little bit of investment to get to this stage, a lot of investment to get to here, but of course as you do that, you reduce the risk. You also increase the value of that asset, and so there's going to be this yin and yang, and we're going to have to make decisions about how far to progress each one of these programs to make it most attractive. And so, put another way, and kind of bringing this down to the Madison level, I recognize, and I'm sure you all do as well, that University of Wisconsin-Madison and the Mortgage Institute collectively make up a world-class basic research organization. And I think there's a lot of rich substrate to help fuel the WARF Therapeutics portfolio in a collaborative way. As Allan mentioned, ICTR is going to provide funds to help support both the early and the late translational sciences. And so to bridge the gap and helping to navigate this preclinical space and effectively connect those basic biological discoveries between the university and biopharma, WARF Therapeutics is going to build a preclinical drug discovery operation, made up of myself, a few other, industry, types that have, experience in navigating this preclinical space. Our job will be to, work collaboratively with the PIs to do the screening and then invest in medicinal chemistry activities to progress these to novel hits from commercially available hits, progress those potentially to early leads, late leads, and ultimately to perhaps even to a clinical candidate. Now, how are we going to do that actually? And so, it's not easy in a big organization. Certainly there's going to be challenges here on campus. Probably challenges that this organization is already understanding. And so WARF Therapeutics has made the decision, we're not going to build internal labs. We're not going to build large teams. And so we need to establish bolt-on partnerships that allow us to build a virtual laboratory. And so we've done a lot of interviewing and a lot of discussions with a number of organizations. And we've decided to spend some of our activities with Sanford Burnham. This is a established organization, as Allan mentioned. They have all of the early drug discovery capabilities that are required from biological assay development, high throughput screening, and they can do those screenings in a number of different formats. They have early medicinal chemistry, 10 internal FTEs and a handful of FTEs around the world. And they've been very successful in advancing novel targets through screening to get around 150 to 175 probe molecules that Allan was referring to in his talk. That said, I've been talking with the cores on campus here, and we certainly want to try to establish a drug discovery presence on campus. And so, talking with Howard Bailey and Mike Hoffman and Steve Swanson, we're going to put together a small team and start planning how we're going to build those capabilities on campus and run a pilot program on campus, trying to move things through this space. There's probably going to be some hiccups along the road. There's going to be some lessons learned. We'll identify what those are, identify the gaps, and try to figure out how we can increase our chances of doing things better in the future. And so there's going to be other instances where we want to move beyond sort of early leads and move things to sort of late lead optimization or clinical candidate nomination. And so we've established we signed MSAs with both Pharmaron and we're almost near completion with WuXi. And these are organizations over in China that have extensive lead optimization capabilities, big medicinal chemistry laboratories, good pharmacology and toxicology laboratories, and drug metabolism as well. And so, this will be sort of a fee for service. We'll, when we need those resources, we'll be able to go off and get those resources. And when those activities are done, then we can return those resources back to them and move forward. So one of the other attractive parts of the Sanford Burnham collaboration here is they don't want to be viewed as a fee-for-service type of organization. They want to participate in a risk-reward partnership. They want to invest in-kind resources to help move these programs forward. And so one of the things that they're really good at is, and it feeds into sort of the incentives to the UW PIs, and that is they're very good at obtaining screening grants for novel targets as well as medicinal chemistry grants. And so they'll collaboratively write the NIH grant proposals with the PIs here on campus, and those programs that get those funds, 50% on average will go to support the program at Sanford Burnham, and the other half of those grant monies will come back here on campus to support the PI's labs to support that program. It also gives, the PIs on campus an opportunity to work with industry-trained, drug hunters and BD professionals. And what's most important to me, of course, is to, relieve, patient suffering and improve and/or cure disease. So, what I'd like to do now is move on to talk a little bit more about the 5R philosophy and the industry-tested portfolio management process. But before I do that, I just wanted to share this graph, which kept many an industry executive awake for the past 20 years. It illustrates nicely the challenges the industry has faced over the last number of decades. And so on the y-axis is new molecular entities or drug approvals. On the x-axis is the year, and in gray is the overall spending in billions. And as you can see, there's a pretty substantial and obvious trend that over between 95 and 2010, a huge decrease, about a threefold decrease in drug approvals. Unfortunately, that was seen despite the threefold increase in overall research spending, it wasn't leading to better productivity. So suffice it to say, it's Allan kind of alluded to a number of the factors: industrialization of drug discovery, going after sort of a shots on goal model, the diseases that we're trying to tackle at this time, oncology, neurodegeneration. Obviously those pathways got much more challenging. We had a lot of biology that we had to uncover and learn. Regulatory climate got a lot more challenging. And so there's a lot of reasons why this happened, but still, this particular situation is certainly not sustainable. And so, despite all of the rationale that went into it, what AstraZeneca realized was that 75% of the program failures was attributed to insufficient knowledge of the target. And so AstraZeneca did a retrospective analysis of their own portfolio and asked, why did our drugs fail, and at what stage? Can this knowledge be applied to improve productivity and reduce attrition? And so they developed a five-dimensional framework that they utilize as a portfolio management tool to help facilitate decision-making and ultimately, hopefully, to improve productivity. And as Allan mentioned, this is based on sort of a 5R framework: understanding the right target, understanding the tissue, the safety. And this is not really about the safety of the molecule in a toxicology sense. It's really, I think, sometimes it's really easy to think about the target and efficacy, but what about the off-target effects of modulating that target? As we're going to see in the JAK2 story, later on, all of the toxicity that was observed in that program was actually mechanism-based tox. And so, there's a needle to thread. And so, I guess the message, to everybody in the room is when you're thinking about, efficacy or thinking about, that target and how it's going to improve disease, we also need to understand, if that drug gets to other tissues and modulates that target in other tissues, what are the ramifications of that? And so, with that in mind, the new portfolio strategy is that AstraZeneca put forward was invest early in basic biology, prioritize those opportunities that have the highest degree of validation, a solid therapeutic hypothesis, deemed to be safe if you modulate that target, and will be differentiated from standard of care. And that gets to the overall patients and commercial potential. The regulatory environment these days is very, very challenging. Gone are the days where we can bring another statin to market. We really need to show that it's going to be molecular or substantially different. It's going to solve some problem of all of the statins that came before that. And so, with any luck, if we do this and we're disciplined to this sort of decision framework, this will lead to, early investment in the program fundamentals, hopefully will, reduce failing later in phase 2 and phase 3. And so this failing early and cheaply versus will allow us to, reallocate resources to other opportunities. And so when we put that, novel, that investment together with a novel chemical asset, hopefully this will provide, the best, licensing opportunity to biotech and pharma. And so a number of people have, talked about this. Robert Plenge, called this type of, process a disciplined approach to drug discovery and development. Ruth, who's a toxicologist, said, in her paper, understanding drug targets, there's no such thing as bad news. If the target is going to be toxic in phase 2, let's figure out by investing in the toxicology, in the preclinical phase, so that we're at least aware of the issues, and maybe we can mitigate those issues. And if we fail to mitigate them preclinically, we better have some other plan going forward into the clinic, because right now about half of the molecules that are failing based on target toxicity. So the impact of the 5R framework and similar systems has been implemented across the industry. And so, and this happened around 2008, 2009. And so, how did things look in the last 8 years? Well, our new molecular entities went from around, I guess we're hovering sort of in the high teens, low 20s in around 2010. And last year we had 59 different new molecular entities that were approved by the FDA. And so almost a threefold, improvement in productivity and only about a 30% increase in research spending. Now, of course, that's way too simplistic a rationale to explain this very, very complicated graph, but at least the correlations and the trends are very promising. So I'd like to spend a little bit of time on describing, the questions and activities and ultimately the data that is needed with respect to the 5Rs. And so, the right target is probably the most important aspect of this process. Understanding the link between, the genetics and human disease. Understanding, is that target for that disease, well validated. And so just to kind of help level set, I put forward this particular definition of target validation, which is the process of physiologically, pathologically, and pharmacologically evaluating that biomolecule. Could be formed at the molecular, cellular, or whole animal level. And so it's really important to understand, the signaling pathways of that target. What about the disease association, the genetics, and the expression data of that disease tissue versus, the wild type tissue? Is that molecular target overexpressed in the disease? Is it genetically amplified? Are there, activating or inactivating mutations of that target that are correlated with the disease? Of course, we're going to need in vitro and in vivo models, and most importantly, I think, is sort of the genetic experiments. Looking at gain of function and loss of function, studies at the in vitro level as well as the in vivo level. This could involve, knock-in or knock-out models, within animals. And ultimately, it could involve, a pharmacological tool molecule. My preference is to delay this, focus in on sort of the genetically, type of validation experiments, whether it's CRISPR, antisense, siRNA, types of, modulations, and, eventually, start to invest in a small molecule. Because by the time that you start this whole process of screening, if you screen a molecular library of 400,000 to 1 million compounds, on average it's going to be somewhere between 30 cents and $1 per well. And so it's a huge investment to go ahead and do that screening campaign. And so if these other genetic tools don't give you the answers you want, it might be a good time to actually rethink that particular molecular target for that particular disease. In terms of the right tissue, we need to understand where does the drug need to get to engage that target? Can we develop assays to assess drug exposure in that tissue? Are there relevant biomarkers in that tissue, whether it's target engagement, whether it's downstream functional biomarkers? And of course, if that tissue is inaccessible in a human clinical trial, are there peripheral types of tissues that we can utilize and build a correlation between, say, plasma and what we're going to talk about on the JAK2 program? The key place was bone marrow or spleen. Once you build those relationships preclinically, you might be able to use plasma as a surrogate tissue going forward. Ultimately, it's really imperative that we establish a clear understanding of exposure, pharmacodynamics, and functional relationships. Right study, understanding all on-target modulations, whether it's efficacy or whether it's toxicology. How are we going to stratify patients in the clinic? Are there good biomarkers that we can do that? How do we know which patients to go after? We've evolved in cancer treatment from chemotherapy to personalized medicine. For example, non-small cell lung cancer now has changed with the advent of the ALK mutation. And so now if you have that ALK mutation, we can give you, crizotinib, for example. And of course, as we talked about earlier, the right commercial potential. We have to understand what is the standard of care of that disease. How is this new molecular target going to improve that? So I would now like to, tell you a little bit about, the program submission process. And like I said, we're going to utilize the JAK2 story as an example, an illustrative example on how to fill out the form, what are the key studies that we're looking for, and so on and so forth. And so, PIs interested in translational disease-related research are encouraged to reach out to WARF Therapeutics 6 to 12 months in advance of submitting your program. Like I said, let's sit down, let's have a conversation. I'd love to set up regular meetings with you and your group to learn about the science, learn about the challenges. Perhaps I can help identify some gaps that could lead to other experiments to help fill those gaps, and then we can plot those next steps. Ultimately, like I said, at the end of target validation, you can submit both the IDR as well as the program nomination form. And you can find that program nomination form at warf.org/therapeutics. And at the bottom of the front page is this particular program submission form. In this particular hyperlink is a blank form. Here's one that actually has some instructions to go along with that. And so you can see here, it's based on the 5 R's: target, tissue, safety, patients, commercial potential. You'll have the name of yourself, the target, the therapeutic indication, and so on and so forth. On the instructional phase are a number of questions that will need to be addressed in this particular document. And so this is meant to be sort of the executive summary. This is just a few bullets that help capture the salient features of all of the data that you're going to present in a series of slides. And so I'm not going to go through all of this. You can read this at your leisure. Whoops. And so after that program has been submitted, I envision this then having a formal presentation to WARF Therapeutics and also to our scientific advisory board, which is made up of currently industry executives, Deepak Delvi, who's pharmacologist and drug metabolism person with experience at both Pfizer and Celgene. Mark Deed, most recently was the chief medical officer at Eli Lilly and the Chorus Group. Chris Dinsmore is currently the acting vice president at Pharma in Boston. Previously, he was executive director at Merck. And Paul Sechrist has experience at both Merck, AstraZeneca, and is now vice president at a startup company in the Boston, Cambridge area. I also want to take time to introduce some of our governance board members. From the UW staff, we have Howard Bailey from Carbone Center. Is Howard here? And I see Rick Moss over here. Rick, if you could just stand so everybody can see. Rick is the Senior Associate Dean at the School of Medicine. And Steve Swanson, I think, is traveling today, but he's the Dean of the School of Pharmacy. And so the governance board is really going to help with the overall strategic direction of WARF Therapeutics. So after the program has been formally presented to WARF Therapeutics, there's going to be sort of a decision and prioritization. And so if the program is accepted, we'll put together a work plan, timelines and go, no-go decisions. We'll discuss and agree on a public disclosure plan. As Allan was mentioning, this is sort of a challenging area in doing academic research. Academia is incentivized somewhat differently than industry. Academia is incentivized to publish and get grants, whereas in industry, we're trying to generate intellectual property and protect those investments. And so by no means are we saying that by working with WARF Therapeutics, thou shalt not be able to publish. That's not what we're saying. What I am saying is, at some point in this initial process, we just need to come together and put together a disclosure plan and timeline. We want to protect the investment, but we also want to do what's right for the academicians on campus as well. And so we'll try to thread that needle to meet everybody's wants and needs. If the program is not accepted, we'll give immediate feedback and rationale. If there are gaps in that data set that might be filled with certain experiments, perhaps we feel that there's a few more validation experiments that need to get done. Allan is going to be nearby, and we're going to have conversations to figure out how we might be able to help you, fund you to get to that next inflection point, so that you can bring that program back and have it reviewed again. So I'd like to use this time to discuss the collaboration that we had with Professor Gilliland at the Harvard Medical School and his discovery of the JAK2 genetic mutation. As I said, to use this as an illustrative example of how we're going to utilize the 5R framework, what data sets are needed at this stage that WARF Therapeutics will find is sort of the most meaningful from our perspective. And like I said, how to fill out that overall submission form. But I'd like to take a few moments and tell you a little bit about the basic biological discoveries of the V617F mutation of JAK2 that Gary and his group discovered. And so, nearly 50 years after Dr. Damchek speculated that there was an undiscovered stimulus responsible for myeloproliferative neoplasms, and urging the scientific discovery community to try to search for this molecular defect, 4 or 5 different groups almost simultaneously in the spring of 2005 reported an activating mutation in the JAK2 gene. Baum-Shanker showed that this particular mutation showed up in around 95% of patients with polycythemia vera, which is, JAK2 is, downstream of the EPO cytokine, and those that are familiar with the biking industry, there's a lot of people that will use EPO as a way to sort of, dope and try to get a competitive advantage. But in a genetic sense, this leads to an increased red blood cell production, a thickening of the blood, increased spleen weight, and increases your risk for, clotting and stroke. JAKs are non-receptor kinases involved in signal transduction. When the cytokine binds its receptor, it leads to phosphorylation of JAK, phosphorylation of STAT, which then dimerize and translocate to the nucleus and drive gene transcription. what's really, really important is that all hematopoietic cells require JAKs for their signaling. And so this gets into the whole safety concern that if our molecule hits all JAKs, JAK1, JAK2, JAK3, and TYK2, you can imagine having a lot of toxicity associated with it, because these processes control platelet formation, your red blood cell production, macrophages, B cells, T cells, and NK cells. And so understanding that is really, really important. And so getting to the 5R framework, the V617F mutation is an activating mutation. The genetic link is it's found in high incidence rates in PZP patients, as well as primary myelofibrotic patients. And this in vitro gain of function leads to constitutive pathway activation, rapid expansion of erythroid cells, and this was confirmed in an in vivo study by doing a knock-in study in mice, and it showed a full recapitulation of polycythemia symptoms. And so to show you the data that supports this, Van Schenker transfected or BAF3 cells with either wild-type JAK2 or mutant JAK2. And in the case of mutant JAK2, you can see that the pathway is constitutively activated. JAK2 is phosphorylated, STAT5 is phosphorylated, but in the case of wild type, this required EPO stimulus for that to happen. They then looked at the growth of those cells, and in the case of the JAK2 mutant, on the y-axis is cell growth, and you can see that constitutive activation of that pathway leads to uncontrolled expansion of the erythroid cells. Whenever possible, performing target validation on patient-derived cells is preferred, and is probably the closest model of disease. And that said, Van Schenker and his group placed cells from PCV patients in a three-dimensional assay and monitored their erythroid colony formation. And you can see when you treat those cells with a control siRNA vector, you see around 150 to 160 erythroid colonies formed. But if you treat those cells with a selective JAK2 siRNA to knock down the protein level to around 10% of normal, you see a rapid reduction of colony formation. And so in summary, this data supports a key role of JAK2 in myeloproliferative disorder and a compelling in vitro target validation package. Professor Gilliland took this one step further. He transduced the V617F mutation into lethally irradiated mice, and what he observed was an observation that these mice, in 3 weeks' time, had all the classic symptoms of polycythemia vera. As you can see here, you see an increased population of CD71, TER119 double positive cells, which are early erythroid progenitors. You can see a substantial increase in splenomegaly and about a 20-25% increase in hematocrit. And so put together, this shows that JAK2 has good in vivo target modulation as well. Just want to take 1 second to talk a little bit about the tissue and safety. So JAKs are expressed not just in bone marrow, but they're ubiquitously expressed, and so we need to be aware of that. It's expressed in bone marrow, peripheral blood, spleen. As you could see from the validation work, there's a number of attractive biomarkers. You have phospho-JAK2 and phospho-STAT5 for target engagement biomarkers. And from a functional perspective, you can look at erythroid progenitors, you can look at hematocrit. So we have a lot of great biomarkers that will be important. But what's also important is what is the effect of prolonged on-target inhibition of EPO? This could lead to anemia. If we dose too high, we might normalize the hematocrit, but we might then drive these patients into anemia. But because platelets are downstream of JAK2 as well. If we hit JAK2 really hard for a long period of time, that could lead to an issue with platelet count and bleeding issues. And so with that in mind, I'd like to then step through a little bit and tell you about sort of where I spend most of my time living, and it's sort of in the medicinal chemistry space here. And so this molecule here has been known as a JAK inhibitor for a number of years. And in fact, it's not just a JAK inhibitor, it hits half the kinome. It's very staurosporin-like. And so not a very good molecule for small molecule target validation. It's too unselective to be able to say that putting this into a cell is going to have a particular effect. And so, my group took a look at the key, we did a crystal structure of this molecule, took a look at the pharmacophoric features of how this molecule bound to JAK2. And so in the various colors, I would say these are the key features that lead to binding to JAK2. And so as we modulated this overall scaffold to try to improve selectivity, to try to improve the drug-like properties, we wanted to maintain these key features, in blue, in red, and magenta. We did this about 5 different times, and so without doing a screening campaign, we came up with around 5 very unique and novel JAK2 scaffolds, and the one that really progressed was that of compound 3. This is now potent, it's selective. The only thing that was a problem was oxidative metabolism at that, this particular spot here, but by putting 2 different electron withdrawing groups on this molecule, that solved the issue. And so very quickly, this molecule is a potent JAK2 inhibitor. It also hits TYK2 with almost equal efficiency. It has good selectivity over JAK1 and great selectivity over JAK3. When we put it into cells, the molecule bound to its target and blocked the downstream phosphorylation of STAT5, and it did so in vivo as well. We then looked at the overall exposure of this molecule, and I'm summarizing a lot of data, but we needed to stay above this IC50 line of 1.2 micromolar for around 20 hours each day. And so you can see when we went from 9 to 18 to 36 mpk, we almost achieved that. And so we actually went to 54 mpk for our in vivo experiments that I'm going to show on the next slide. And so what we wanted to find out was, what I need to say here is that we actually put a bioluminescence tag on the V617F allele so that we could actually quantify in a real-time way, in a non-invasive way, what is the copy number of that allele and where is it accumulating. And from this particular, the vehicle-treated mice, you can see, high concentration of the mutant allele is accumulating in the spleen. And so we asked the simple question, how many days of dosing of 54 mpk of compound 4 is required to reduce that allele? And you can see the answer is 3 days. And so the color intensity in the spleen is gone after 3 days of dosing. So that's the good news. The bad news is on-target toxicity. 3 days of dosing of this molecule led to a rapid depletion of NK cells. And so the risk of opportunistic infections was really, really high. And so we then said, okay, how long is it going to take for the NK cell population to rebound? The good news was around 4 days. And so within air bars, we've repopulated all of our NK cells. And so what we then asked was, how much did the V617F allele rebound in those 4 days? And you can see it did rebound, but it's less intense than it was on day 0. And so we asked the question, could we do a 3 days on, 4 days off dosing, and would that be safe and efficacious? And the answer is yes. And so this summarizes an awful lot of data. And the important thing is the hematocrit. After 2 weeks of this 3 days on, 4 days off, we've normalized hematocrit. This particular column right here is what the hematocrit of a normal class 6 mouse is. And you can see we started at this level here, And by 2 weeks of that dose and schedule, we've normalized the hematocrit. So that's sort of the data package that I think is going to be most profitable for WARF Therapeutics. Something to keep in mind as you're bringing forth programs. And I just want to sort of reiterate, we want to collaborate with campus. We hope this is going to be a great and productive relationship. We're looking for PIs with well-validated targets, great therapeutic hypotheses, and want to collaborate to move therapeutics in areas of high medical need. And so with that, I'd like to thank my WARF colleagues listed here. Too many to mention, but this group has been really instrumental in getting me acclimated to WARF, getting to meet many of you on campus, helping to message the overall vision of WARF Therapeutics, and then there's Janan and her group to help put this program together, build our website, and help get the WARF Therapeutic message out around campus and across the world. And I would like to thank you for attending, and I'd like to invite Allan up, and we'd be happy to answer any questions. Yeah, so if you have a question, please raise your hand. Somebody with a mic will stop on by, and I just ask if you could state your name, what department you're in or what company you're working for, before you ask your question. Thank you.
Brian Johnson, biomedical engineering at UW. So I saw a little bit of conflict in the talk, and it was based on that phenotypic identification of a target, right? And it's really in conflict from what the standard drug development protocol is, right? We need to know that molecule. And if you do that, you really eliminate functional assays and a lot of things, right? So in your JAK example, if you started with that colony-forming assay, right, that patient cell line, and you screen that to start with and identified, would you find that target? How do you see that approach? Is it in conflict? Is it just a different way to think about drug development, and is it tenable?
I think that's a great question. And I think my view on that has evolved over time. At Merck, we kind of, looked at things from a hammer and nail perspective. We thought that everything was could be solved from a small molecule. And when we went into clinical trials, we were unsatisfied if we couldn't answer that little box on clinicaltrials.gov of what the target was. But going back to the Revlimid example, patients were dying of multiple myeloma, of that acute disease. Patients benefited from Revlimid. It took 6 years for them to figure out what that mechanism was, and that's going to inform the whole PROTACs area going forward, which we can't really talk about today. So I look at molecular screening and phenotypic screening as complementary. I think there are two very nice approaches that are should be considered. It's a great question. Allan, do you have anything to add?
Yeah, thanks. Vince Crines from the Department of Medicine. Could you just give us a few more details about the pilot? Is there, is there a rolling nomination? Will there be certain deadlines? Will there be a certain amount of funding for those pilots, or is that all individualized?
So when you're saying the pilot, you're talking about the on-campus pilot.
Yeah.
So I look at that as, a separate, or a complementary work stream to the activities we're going to be doing with Sanford Burnham. And this is based on a detailed analysis of, the capabilities that we're needing to play in the early drug discovery space. I looked at Sanford Burnham and all of the necessary capabilities, and what's really important and was often overlooked is, sort of the sample management and all of the bioinformatics that, go behind that, and the databases to store the data and to firewall that data. And so all of those processes are, already available at Sanford Burnham. And so right now at UW, they're not fully quite where they need to be. And so it's going to require a little bit of experimenting to figure out what are the gaps in terms of some of these capabilities from an IT infrastructure perspective. And so I look at, where we do the work is less material. To your question about funding, I think it's going to, the funding mechanism is going to be, what is necessary for that well-validated target that WARF Therapeutics and the Scientific Advisory Board, want to fund. The key questions are, how much do we have to invest to get that first go, no-go decision? And so I don't think that there's going to be one simple answer to that. It's going to be based on, the time and the types of activities that need to be accomplished.
I think, Vince, the other side of this is that there are some ICTR pilots that you might think about. This is to get you up to the point where that you would satisfy the 5 Rs. So we have an annual cycle for the ICTR pilot grants. These are the novel methods type grants. We're also rethinking the Early Pathways to Commercialization Award. And these are much smaller grants that have been awarded to U of W PIs to help advance along the translational science spectrum. So I think what we're going to be looking at is reformatting that pilot grant to be focusing on developing the preliminary data that would satisfy a rigorous target validation, being able to satisfy that you have a relevant animal model, that you've done the study in a way that's rigorous and reproducible, that you have genetic evidence and or perhaps identification of a biomarker. So we're still in the process of negotiating and thinking about what sort of things we're going to support on that, but I think over probably the next 3 to 6 months we'll have a plan, and you'll hear about this through the regular communications. My guess is that it will be best to have this on a sort of a non-annual, but more of a nimble sort of way, maybe three cycles, 3 review periods a year. But we have yet to develop how that looks. I'm not sure if that answers your question.
Hi, John Yin here, chemical and biological engineering. I pursue systems biology and systems chemistry here in the building. So the focus was mainly on targets and how to validate them at a greater depth. Once you have them, you can do your screens. But the therapeutics could also refer not only to small molecules, but also potentially to biologics.
Sure.
I wonder if you could say something about your interest there. It seems like the panel and your experience is more on the small molecule side.
Sure, I think that's a fantastic question. Certainly, I think, like I said, after 20 years at Merck, we thought everything had a small molecule solution. If you look at Merck today, their best-selling drug right now is Keytruda, which is a biologic, and so they're a little bit slow to adapt to other drug modalities. What I want to point out is, while I was at Celgene, I spent most of my time working on PROTAC modalities, which is a very different way to, other than small molecules, to, essentially affect, modulation of that disease-relevant protein in a cell. Once I left Celgene, I then worked at an siRNA or in a microRNA company, and I worked on oligonucleotides. And so I think about antisense, siRNA and microRNA, agonists or antagonists are other modalities that are going to have potentially preferred status for a given target. And so I think peptides or biologics are other good modalities. And so I prefer, and yes, most of my experience to date, 80% of it is small molecule, but 20% of it is in this other category. I want WARF Therapeutics to be modality agnostic, and I want us to recognize, and I sort of had on, the 5 R's. Lee always gave me a hard time. It's like your 5 R's is actually 6 R's, John. The right modality. It's like that was my addition, because I really want us to understand, a biologic, of course, is not going to be great for an intracellular target, but for a cell surface target, that might be preferred. You might be able to eliminate some of your off-target activity, if you prevent your molecule from going to certain tissues or getting in cells. And so I think we need to look and say, what is the right modality for the target that's being proposed in that disease? Thanks.
Can I follow on the questions and with some extensions? So how about the delivery methods or delivery modality to better deliver the therapeutics?
So that's a great question. During my last year at Regulus, I quickly learned that coming up with an oligonucleotide molecule was the easy part of the solution. But trying to get that oligonucleotide, anywhere other than, the liver or the kidney was a challenge. Getting it to, the lung or getting it to the brain was a challenge. And then once it gets there, how does it, get into the cell? Oligonucleotides, were designed, the barrier was designed to keep them in the cell and not leave or enter the cell. And so I think delivery is really important. But I think it's going to be outside the scope of what WARF Therapeutics is going to focus on right now. I'm hoping that, if we take a type of modality forward that is going to require delivery, I think we need to understand and recognize that early on, and recognize that we're going to have to invest in delivery. But I think WARF Therapeutics is going to focus on sort of the molecule and the target, and bring in delivery platforms, if necessary. I think we have time for 2 more questions. John.
So I'm John Deneau from WID and the School of Medicine and Public Health. Maybe this is a bit philosophical, but if you have some tangibles for the WARF Therapeutics answers on this, I'd appreciate it. It seems to be in your presentations, the biggest issue we face and pharma faces is target validation. I mean, you both were pointing to the fact that's where pharma has failed in the past, perhaps. And, it's not, and you pointed sort of to academics as filling that gap. And I would say that I think that's partially true. I don't think we quite have the funding mechanisms, say through NIH, that provide the level of funding that would be required for target validation. And my experiences have been with pharma that target validation is way deeper, way more extensive than what a typical academic researcher is capable or typically does. So how does one fill that void if, say, pharma's looking to the academic researcher to provide to fill that gap, and the resources aren't typically there, or the incentive typically is not there?
Well, like I said, we were thinking about this might be an opportunity for ICTR to help identify and provide some support for some of these early biological validation type studies. I agree with you that this is not something that is typically supported by the NIH mechanisms. So I think that this could be an opportunity for ICTR. We will probably have to develop some partnerships to be able to support these types of things. But we were thinking, again, like I had mentioned in response to Vince's question about coming up with a program for biological target validation with the idea that this would be bought in by WARF, so that if we go through and do these studies, if we meet this bar that is needed for biological validation, that program would then be of interest or acceptable to WARF. So we're still working on trying to work out how that.
Sorry if I missed that answer.
Work.
I had to go.
No,
I had a
that's
bio
fine.
break.
Okay,
I'm
all
just-
But John,
sorry.
that's a very important question.
Yeah.
And
So we have to figure out a solution to it.
And this is something I circled as soon as I got here. And I do believe that funding for target validation, even take the JAK2 example from Professor Gilliland. That was about a $250,000 in vivo experiment to do that transfection into those mice. But it really gave you the confidence that that particular mutation was in fact the real driver of that molecular disease. And so when you think about that $250,000 from a target validation perspective compared to maybe $1 million to do a screening campaign and another quarter to $500,000 to work up those hits and identify them as leads, all of a sudden $250,000 gives you a nice insurance policy to make that next investment. And so I think we need to find a way, and Allan and I will continue to talk about ways that we might be able to, help, fill some of those gaps. And you're going to hear more about that in the future.
I mean, I think there might be, you guys might figure out that, say, 75% of the targets amongst the folks here could be validated through a similar mechanism. So that could be something, right? You could invest in a pseudo high throughput way of validating targets that all, would occupy or would satisfy a number of investigators here, for instance.
Sounds like you want to chair the,
No,
the committee for target.
Well, if you have ideas, be sure to let us know.
I'm just thinking out loud. So
That would be a great solution.
We'll accept your nomination, John.
here, thanks.
Terrific. Well, I think we're going to go to break, but let's step outside, have some coffee. If you have any other questions, please come by and talk with us. I want to highlight John Nagel and Rafael Diaz in the back. They're going to have a table outside. These are our business guys from WARF that will help you with any business or licensing questions. And so please stop by and say hi. Thank you very much, everybody. And at 11 o'clock, we're going to start our panel discussion. So please be prompt. All right, I think we're ready to get started here. I'd like to introduce our panelists here. And so, as I mentioned earlier on, Doctors Jennifer Golden, Luigi Puglielli, Doug McNeel are all working here on campus. They're at various stages, in their drug discovery, adventures, and hopefully we can learn a little bit more about their experiences over the next hour or so. So what I'd like to do right now is, ask them each to, sort of introduce themselves, take about 5 minutes, give us your background and a little bit about your research program, where you're at. Luigi, do you want to, get us started?
Sure. So I'm Luigi Puglielli. I'm a professor in the Department of Medicine. So I'm interested in the molecular mechanism of underlying both physiology and pathology of the brain as a function of both development and aging. And so a few years ago, 10 years ago now, we stumbled into a biological process that was not expected to occur. And that led us to discovery of a acetylation machinery that specifically works in the lumen of the endoplasmic reticulum. And soon after that, we discovered that it regulates proteostasis, protein homeostasis with the organelle. And then we discovered that it was relevant to disease of both development and aging. And this spanned from autism spectrum disorder to cognitive decline that normally accompanies aging as well as Alzheimer's disease. And so with the help of WARF, with the lead initiative, Laura was heading at that time, we went on into drug screening. I don't have the expertise of drug screening as suddenly I was doing drugs. We identified compounds, we did some preliminary analysis, and then there was the Z Pharma Station here, which is led by Ed Elder, and the of the small molecule facility led by Michael Hoffman, we were able to identify a few compounds and move that into the mouse. And then we showed that these compounds were able to rescue an accelerated form of aging that we have uncovered in the mouse, as well as was able to rescue Alzheimer's disease in the mouse. And now we are trying to push that forward.
Terrific. 5 minutes? Perfect. Well done, Luigi. Jennifer.
I'm Jennifer Golden. I'm an assistant professor in pharmaceutical sciences within the School of Pharmacy, and also a affiliate faculty member in the Department of Chemistry. So my background is a synthetic medicinal chemist. I was trained as a synthetic organic chemist and spent a number of years in industry doing small molecule drug discovery, doing lead development. I was at Amgen for several years before I was recruited away to help establish an academic drug discovery center where we did a lot of hit to lead and lead optimization work. And so I worked with a great number of biologists across the United States, as well as with screening centers, some of which were described today by Allan and John. So I was part of the Molecular Libraries program sponsored by NIH. And then since then, I had established my own independent group here at Wisconsin. And so I have been working in the area of anti-infectives and anti-cancer drug discovery. And so in my group, we're very interested in making drug-like molecules by innovative means. And in doing that, we generate libraries of compounds that we send out into the world or across campus. Very interested in getting those molecules, just a little plug for our library that we've generated here on campus, that if you're interested in screening compounds, we would love to get those into your hands, where you can look for activity that we would be more than happy to work with you to develop. And so that's kind of in a nutshell what we've been doing. Our flagship project is focused on antiviral therapeutics. And so right now we're pretty excited that we have compounds which came from a screening program and have really matriculated to the point of preclinical candidacy. And so we have advanced leads. We're excited to move forward as a potential clinical candidate here down the road. It's like this. Yeah.
Excellent. Doug.
My name is Doug McNeel. I'm a professor in the Department of Medicine in the section of medical oncology, hematology, medical oncology. And my lab for about 20 years has been interested in immune-based treatments of prostate cancer. So typically when patients are diagnosed with prostate cancer, they have the gland either removed or irradiated, and about a third of patients will have the disease come back, and it's usually heralded with a rise in a blood test that we can detect. And so our thought a number of years ago was, well, if we could intervene with a therapy to elicit an immune response to the prostate at that early stage, we could perhaps eradicate it early and not have the disease come back. And so we set up, we were interested in looking in patient samples of what was immunologically recognized in the prostate, and then in mouse rodent models looking at how do we immunize and get rats or mice to reject prostate tumors. And where we were 20 years ago was with the thought of if we had a simple way to immunize people, and we landed kind of on using DNA as an approach, that we could basically swap out the antigen of interest. And the approach the thought 20 years ago was if we could use a simple approach and sort of investigate the targets in a number of early phase clinical trials. 20 years later, we've now looked at two, and there are a lot of barriers to getting things into clinical trials. So it was a little bit naive and perhaps optimistic 20 years ago. But we entered things into clinical trials, and things started to shift focus a little bit, where now we are starting to see immune responses. We're starting to see some changes that make us think, well, this is something that actually should be developed. And so in 2012, we launched a biotech startup company really focused on the clinical development to take a look at this as a product that we might actually pursue, while at the same time the research lab has continued to explore the basic biology mechanisms. And I think that's been a good partnership. So over the years, it's been interactions with WARF and multiple funding agencies.
Terrific. Well, thank you very much for those introductions. The way I think this is going to work here is I've got a number of questions. I could ask questions all hour long, which is not what I hope to do. So I'll kick things off with a few questions, and then open it up to the floor. And like we did in the previous Q&A, if you got a question, please raise your hand. Somebody with a microphone will come and hand that to you, and we'll go from there. So the first question is for each of the panelists here. Perhaps we can start with, we'll go the other direction here, start with you, Doug. So what are the unique barriers and challenges of doing translational research here in Madison in general, but more specifically on campus?
Probably already heard, and I wasn't present at a lot of the meeting this morning, but the major barrier is cost, frankly. So there are costs for laboratory research and there are costs for clinical research, which are an order or two greater than the basic costs. But there's that bridging the gap of how do you get things one place to the other. And I think that's traditionally always been the hard thing to figure out. I think also there's been a changing climate at the university in a positive way. Translational research was viewed as less desirable if it was, if you're taking basic principles and applying them, maybe that's not as important or relevant to the university as more basic fundamental research. And I don't agree with that. And I think that's an attitude that has been changing over time on this campus.
Terrific. Jennifer, anything to add?
Yeah, I would just say that from my perspective, some of the barriers that I've encountered with different folks who've come to talk to us with my other hat on in the Medicinal Chemistry Center is probably navigating partnerships and also finding resources. And so I think there's been a huge initiative across campus, and actually this was highlighted in Allan's talk and his slides, where Isabel, Gerard, and others have really come together with different cores and finding a nexus through which investigators can find people and find resources, and just having an acknowledgement of what you can plug into and how you can get things done, I think is really valuable. And so there's been a huge push to kind of get that information out amongst other people who might use it.
Excellent. Luigi.
First of all, barrier is funding. I agree with that. So my research is funded by 3R1s, and I have to fight to get those 3R1s. And I remember I sent another one to NIH because I wanted to do a screen out of WARF, and it got a 53 percentile because it was too risky. And so there was WARF there, and we were able to do the screen. I sent back with the data of the screen. It went back to 2 percentile. So, and that's the first time I stumbled to this issue, and then I had the problem recently where the NIH told me this is too risky and to wait a year. Went back, and the grant went to 4 percentile this time. So there is always this fight. The NIH wants mechanist, and I have to try to make a little bit of translation within the other one so it doesn't damage the rest of the package. So that's one problem. And obviously, we need support from the university to be able to do that, and there are mechanisms for the university to give that. And I did receive money from the university to run that, and so I was able to survive that stumble. The other problem is that I'm interested in molecular mechanism of disease. I'm interested in translation, but I'm not a translational guy. And so there is infrastructure here that helped me through this path. We went to WARF because I want to patent the targets. We identified the targets, done the biochemistry, and WARF told me, you can't patent nature. But Laura immediately saw this and said, this would be very good for a screen. And so WARF actually paid for the screen. We did a screen, and suddenly I was doing drug discovery. And so we managed to move through the help of the university and then other foundations and grants to do. And now we have 4 compounds that, when treated in the mouse, can rescue an accelerated form of aging, but they can also rescue Alzheimer's disease. And so now I'm at the point that really with 4 compounds, these 4 compounds we know that bind more or less in the same pocket of the enzyme in the structure, but have some differences. And so now I'm at a point where we could actually look at these compounds to see what is the the common scaffolding unit of these compounds, and that's something that I would like to do and modify some of these compounds, and I don't see a lot of that help in the universities. That's probably a space where the university and perhaps WARF could invest by a system that you can actually do, use these compounds, see whether there is a scaffolding unit that is a common matrix, common unit of all these compounds, try to do modify, and then we have a great expertise to do in silico screen. We try to do that, but we didn't have, we were not successful. Even there, the university doesn't have all the infrastructure to actually push in that direction. That's something that I think is worth doing.
In silico virtual screening is complicated on a good day, in big pharma as well. So that is a challenge the whole industry is facing. But I think the mechanism that your group uncovered, I think, is a little bit transformative in my opinion. When I think about the other mechanisms of neurodegeneration rescue that have gone into the clinic, whether it's gamma secretase, tau, and beta secretase, this mechanism I think is going to be rather transformative, and I'm really excited to see where that research goes going forward. One more question for everybody, and it's more of a philosophical question. What fuels your translational or basic research? Is it scientific curiosity, the challenge, or is it altruism? Wanting to help fellow man and woman to try to alleviate pain and suffering, or is it for financial gain? Wants to start?
I've been nominated by the panel to go first. I guess for me, it's definitely a challenge. So we tend to pick problems that are going to be kind of new and innovative. I would say it also is altruistic. We tend to work on rare diseases, and so these are not necessarily problems that are being followed up on by pharma. And so there we think we can make a significant difference. If I'm going after financial gain, I'm probably in the wrong place. So I should probably go back to my pharma days. But so no, I think that there's great stimulation in going after a real problem and being able to apply one's skills to successful.
I'd echo some of those things. I'm a medical oncologist, so I see patients with prostate cancer on at least a weekly basis. So a lot of our research is fueled by, well, it's nice to discover something in the lab, and that does. I mean, that's why I ended up in as a faculty member. I mean, I'm interested in basic principles and in understanding and pushing the field forward. But as a physician, that changes you in the sense that you also now want to see things happen in people, and you want to see this research make meaningful differences. So anything we discover, I mean, the questions are how do we, how can we learn from this, and how can we adapt this, and and is this something that's going to be translate forward?
Yeah, that's a great story, and it reminds me of the interaction I had with Professor Gilliland at Harvard. He's also a hematologist, saw patients with polycythemia vera, and so he directed his research in an area to help the patients that he was serving, and so he had this altruistic viewpoint that he wanted to have a meaningful impact, and so when it came to all aspects of moving that drug forward into the clinic from a publication type of strategy, he's like, John, my motivation is my patients. That's my goal. And so, I'm not surprised to hear your story there, Doug. Luigi.
So it's a combination of both. The PhD part in me likes the curiosity more than MD part of me likes to some answer to diseases that we can tackle the symptoms, but we can really prevent or cure. And so when it comes to neurodevelopment or neurodegeneration, we can make a very nice diagnosis, but we can give an answer to the patients or the families that really they're looking not for the philosophical, mechanistic part behind it. They just want an answer to it. And so combination of both. Certainly not the mind.
Terrific. So are there any questions from the audience? Always starts this way. Nobody wants to be the first to raise their hand. There we go. Please state your name and your department if you could.
Yeah, my name is Derek Belt. I'm in the Department of OBGYN. And so we work in the area of pregnancy and hypertension in pregnancy. And there's a lot of fear, I think, in drug discovery in pregnancy. And even if you haven't worked specifically on that, I wonder if any of you have worked on a vulnerable population in the space of drug discovery in a population that there's a lot of fear.
Well, certainly that's, dealing with Alzheimer's disease, that's not an issue for me. And some of the diseases that we tackle are early development, but early is postnatal. And so, again, concern for our targets, yeah.
I guess I would just add, there's probably people here, well, in fact, there are people here who can speak to this better than I can, but I would just add that when you get to the point of where you're thinking of your molecule or whatever modality it might be, of really going into any population, and especially those most vulnerable, thinking about infants, I looked in the areas of respiratory syncytial virus, and when you're thinking of treating those very young patients, one pauses, right, in terms of safety and the bar that one must pass to do no harm, essentially, and really hope that your therapies are doing what you hope that they can do to help a situation and not cause any more. But this, I would say there's fear not only in the population of the patients, but also on the side of those who are coming up with new drug targets and discoveries and pushing them into that population.
And industry trying to pick up anything that comes out for yourself and trying to get the compounds into the hands of industry collaborators, and they don't really want to touch it.
So most of my, the last 15 years of my career has been in serious diseases, like oncology, and so the barrier to the questions that you're asking are absent there. But in my previous 10 years, working in autoimmune disease and so on and so forth, you're working in a otherwise healthy population, and so I wouldn't say that we fear working in those populations, but we're certainly it gets back to the question around safety and the 5R concept. And we really need to set an exceptionally high bar. And we put in tirelessly and comprehensive processes in industry to look for mutagenicity, to look for reactive metabolites, to look for key pathways that might be triggered and lead to harm in that otherwise healthy population. And so I wouldn't say it's a fear, but it's certainly heightened our level of rigor in those patient populations.
It's actually one thing that in some ways is an advantage being in the university as opposed to industry. I mean, we had specifically wanted to investigate therapies, and it's not really, it's not the same kind of risky population, but in men with early recurrent prostate cancer who are otherwise healthy. And that's not an area that industry's looked at because it's such a long road to an approval process. But we thought, it's really the right place to be investigating these kinds of therapies rather than people with very late stage disease where industry's focused because they're really, trying to get a very quick endpoint to get a drug approval. And so I look at it as, we have the opportunity to, I think, do the right thing in investigating these therapies where they need to be positioned.
Jennifer, so perhaps you can elaborate a little bit. Having spent time in large pharma like myself, what do you see as the unique opportunities in academic drug discovery? How does your industrial experience, influence your academic research? And how does it influence your teaching and maybe recruiting advice you give to students?
Oh, really good questions. So I would start off by saying, the industry experience I have has greatly influenced what we do on a daily basis. It's a lens that I apply to pretty much how I look at teaching, as well as how we choose the projects that we work on in our laboratory. So I think with respect to the first part of that question, which is, how are things different per se? I would say that, you come in every day in an industry position with a very clear focus of what is going to advance a drug potentially for the benefit of patients and how that benefits the company. And I think there's a little more latitude in academia to maybe go down some of those rabbit holes and follow up on chemistry and chemical biology that I think goes to some of the things that were already talked about in the slide decks, which is where we can contribute to target validation. And I would just say chemistry can help with that too.
Chemistry is usually the solution.
Yeah, so thank you very much. I didn't pay him to say that. So I think there's some differences there. Certainly the freedom to operate is really worthwhile in academia. And then with respect to teaching and graduate students, so a hefty portion of my teaching load is focused towards the future pharmacists, so those individuals you'll be seeing across the counter or in a hospital setting. And I think it's really important to illustrate the whole process. So they're kind of focused on the end game and how they deliver the appropriate medication to a patient. But I like to take them kind of back to the bench side and say, these are where these things came from. And they don't have a whole lot of interest about that in truth. But the middle part is really important, which is why did the drugs evolve to be what they are? And when you have a cross-section of drugs to choose, how do you choose the appropriate one? And that really kind of goes back to pharmacology and chemistry, right? So I love to give them those anecdotal pieces and kind of give them that kernel of knowledge so that they understand when they're pulling that pill off the shelf, that they understand kind of where that came from and why it might be the best choice for their patients. And then certainly I've already talked about our research, which is I could talk about that all day, but certainly we like to choose those projects where we think we can greatest influence how we move forward and how ultimately help patients.
I want to pick up on something that you mentioned, the rabbit hole. I think that's something that's been missing in industry for a long time. We can't afford to go down rabbit holes. We can't afford to take time to do just basic research anymore. It's very industrialized drug discovery. When I joined Merck, they said, spend 20-25% of your time doing novel, innovative things. Well, quite frankly, 25% is not a lot of time to do much, but at least it was something. And some of the greatest programs that I worked on in the early 90s came from that 25% effort. Gone are the days where we can afford to do that. We talked a little bit about drug pricing and productivity, and right now it's, we'll choose the targets, we'll choose the direction, please do as you're instructed. And I think we've really lost our creative influence. And I think this is where academia still, to some degree, can really take that latitude to explore off-the-wall ideas. And from that research, I think can come great things. So go down those rabbit holes.
I hope so. We have a project that's would have been canned in an industrial setting for sure. But we are where we are because we went down the hole. Yeah.
Terrific. So Luigi and Doug, I think you've addressed this question a little bit, but maybe you can expand upon it if there's anything left to say. But I'm really interested to hear how your clinical practice has influenced your basic research activities.
Well, for me, it's simple. I used to be an MD, but now I'm not using the MD, so the clinical practice has gone away.
I think I already kind of addressed it, but yeah, so I'm an MD 1 day a week and then a PhD. Okay. So the 1 day a week influence is what we do 4 days a week.
Michael.
Thank you. It's Michael Falk from WARF. I was excited 15 or 20 years ago when we sequenced the human genome, and I thought it might help us find new drugs. Didn't seem to. Were you excited when it happened, and do you think it's true it hasn't helped, and will it?
Well, I'm a biochemist, mechanistic guy, so the gene doesn't tell me much. So there is always a barrier to, you have all these genes, what are the proteins doing? What pathway, what is the biochemical activity of these pathways? And how these affect in this framework of physiology and pathology. So the gene was not for me, obviously important in the achievement, but was not something that automatically I was associating with new therapies.
Or at least for our work, it hasn't.
Further afield than these guys are, but I'll just give my 2 cents that I think that was the start, at least from my perspective, at looking at broader issues within chemical biology, and so that was maybe the first step, and everything that's come after. So today, we're looking at CRISPR technology and all of this. I think that it set us on a path, right, to ask bigger questions, perhaps. So maybe that particular technology or advancement in of itself wasn't piloted in a way to make drug A, right? But maybe the follow-on technologies will be pretty influential.
I know I'm not on the panel, but maybe I'll give my 2 cents. I think it was met with a lot of anticipation and excitement. But I think it was actually part of the issue with the decline in drug productivity. Like I said, Merck had a medicinal chemistry group of 1,200 people. That's, fully funded programs. We can work on 40 or 50 things at a time. And so what the human genome allowed us to do was, we didn't slow down to validate those targets. We had the medicinal chemistry capabilities to work on 40 things at a time. And there was this belief that, let's validate our targets in phase 2B. We're good at making small molecules that are potent, selective, bioavailable. Let's do that and ignore the validation aspects of drug discovery. And we've made a lot of potent and selective kinase inhibitors that have absolutely done nothing from a pharmacologically relevant perspective. And so I think what we've learned in the 10 years since this rethinking of our strategy is to we really need to focus on, yes, we've uncovered a lot of targets, but now we need to really tie those in a very intimate way to molecular disease, that molecular property to the human disease. Any other questions from the audience?
This would be one towards, tenure track problems and the conflict between what WARF would want and delaying publication and patent. Do you guys feel in your departments that you're, that could damage your tenure ability to sort of delay those things? And do you think it would be valued, or is there an emerging appreciation for that kind of work at a department level?
I've heard that issue, or I've heard that raised before. I don't know that we've ever slowed down any publication for patent protection reasons, and some of that we've let some things go that we didn't think about. But also we've, when interacting with WARF, they'll actually speed up the whole process to make sure if you have a manuscript together, that helps them out in terms of putting together provisional application and things. So I don't know that we've ever, say, slowed down a publication for that reason. It was never really an issue. I never had the feeling like I was, we've always had scientific questions going forward. Even when we did a toxicology screen, we could either use Covance, which would have cost us 6 figures that we didn't have, or we could do it in the lab and we cobbled money together, did it that way. That way we could publish the results. We've always been kind of, academically minded, and that works for both.
Yeah, same here. I never had to face that issue. I mean, probably where I see where you could find the problem is when you find a molecule. Like in our case, you test it and everything is ready to go, but it came out of a screen. And so obviously for WARF perspective, if you do modification and you optimize that, it's much better than just sending out the compound. But you know, that's what we did. But I didn't see a pressure.
Sad that we're kind of at this point right now where we've made some compounds that are very interesting. We're excited to see what they're going to do next. And so it's been a very careful timing issue between patent execution and publication of our results. And you know, I benefit greatly from probably the past experience I've had between 15 years of academic and pharma drug discovery experience of being aware of that in advance. And you can't always time these things that expertly, right? But it's kind of working out. So, so I think that just having a conversation is really important. That transparency of if one is in a position to have compounds that one wants to protect and the other parties are willing to protect it. I have found that there's great flexibility in the timing of that and acknowledgement of the papers that have to come out around that and just going out and talking to the world about it as well. So far so good, but I would just admonish that, you know, I think that having those conversations as early as possible when you think you might be coming up on that possibility with WARF is a good thing to try to mitigate any issues.
Is there anybody of my WARF colleagues from the IP side that would like to chime in on that from a timing perspective?
Patrick again with Arceus Technologies. I'll share this experience. So we recently licensed technology from WARF, and a publication was in process, and the WARF folks were very supportive in accelerating the patent filing to get out ahead of it, to avoid any prior art. So we had kind of the opposite effect, where, I mean, there was some interesting, it was important to get the publication out, but so the WARF folks that accelerated their end so that we could try to avoid any prior art.
Yeah, that happened with me as well.
Terrific. This will go out to anybody on the panel that has some ideas. But are there any activities that could help foster a stronger drug discovery culture or mindset on campus?
Probably just go back to saying, a couple things that pop in my mind are, navigating partnerships external to the university. I think we're always looking at ways at how we improve, how we might do that. And how we make that go maybe a little faster. Because really all parties have to come together in an agreement that compounds or biologics can go to the appropriate people and be tested. So that's one of the things I think that comes up.
Well, I'm not a drug screener of person, and so I've been navigating this with my MD background and with my desire to find mechanism, and because of infrastructure in the university that has actually pushed me into translation. And so suddenly I find myself working in translation, and even though I'm not trained to do translation, I think that speaks highly of the university in the sense that there are systems, and there is enough infrastructure that can help you go into move from more mechanistic target identification into actually translational research. There are some points where I had some trouble, but we were able to outsource this to outside companies. So I think overall it's the infrastructure is well positioned, and I think that the WARF Therapeutics initiative is probably going to help bridge more with the pharma world. This is where I actually have most of the difficulties, because as soon as we publish the mechanism of the first compound, I start having phone calls, and they asking me to do this experiment, to do this. Have you done that? Have you done that? And I say, I'm in academia. I'm not developing the drug. I am interested in mechanism, and so that party, said, I think, to me when we met the first time. In the old times, you would identify something, you would license that and give it to the company. Now the pharma company won't expect you to do more, and that more is probably where we're gonna need more help, because we are not trained to do that.
So I apologize for our first couple of meetings when I asked you if you've done this and have you done that. And
that didn't come from you. It came from the phone calls.
But hopefully, not only will I ask those questions, but will help you actually do those particular activities that will be necessary to build that data set, to bridge towards biopharma.
Yeah, I've learned a lot.
That's great. Yeah, I think a lot of the elements are there. Sometimes it's finding these resources, figuring out how to make some of these things work. We were in a little bit different situation where we early on wanted to have, we wanted to push things into early phase, phase 1 clinical trial, and we got funding to do that and pushed it in. And that was a little bit different because then we had to, once it was already then tested in a phase 1 trial, it was difficult to then have that enter and get licensed out without a lot more data. Once you've already de-risked it, and from a safety-wise, then now pharma came back and said, well, we'd like to see results from phase 2 trials. And so and then it was a little bit different. So if we're going to pursue that route, now we need to then be really setting up a company that's really working on that. And that was a little bit challenging to kind of figure out how to make that work within the universities.
Yeah, so along those lines then, Doug, so, you've taken that molecule, into the clinic. You've navigated that, those hurdles. Any lessons learned that might influence your approach, and will you, be interested in going down that path a second time?
Well, I think we're in some ways already starting down that a second time, because we have something else that's now started in a phase 1 trial. And we're I think you learn from all of these experiences along the way. I've been asked a number of times, what's it like to be an entrepreneur on campus? And it's like, I'm not an entrepreneur. I'm just trying to get my work done, and it's just trying to figure out how to get that done. So.
Terrific. Anything else? Yes.
Hi, I'm Kathy Rasmussen, Assistant Director for the Forward Bio Institute here on campus. And you touched on things that you had not been trained to do. And you talked about what you learned in the process. And you mentioned talking about how you're actively training pharmacists. So what about your experience are you bringing to your graduate students and modifying the training programs you're doing to prepare the new PhDs who are going to go into a completely different paradigm of R&D per your discussion?
That's a great question. I've noticed over the last 10 or 15 years, the reality is 17% of our graduate students will end up in academic positions. Already are ending up in industry or other areas. I'm having more graduate students coming to me that say specifically, I mean, they all used to say, oh, I want to go into academia, and then you knew that only 20% of them would. But now they come in because they specifically want some experience where they see translational research, or they want to have that kind of area where they can do basic research, but also see how this might apply for, about more applied sciences. I think the students are starting to ask those questions.
I think that for me, I kind of fulfill a certain niche that maybe wasn't here before, standing with one foot in academia, one foot in pharma. And so the students actually come, even when we're recruiting, and they want to know, what's my life going to be like over here? What's my life like going to be here? And what choice should I make today? And so I like to give them a little bit of perspective. They do ask this question, is like, where am I going to be happier? And it doesn't really work like that. So I think, kind of setting the stage for giving the real world ideas about what the challenges are on both sides, and it really comes down to a personal preference and where your skills lie. And so I think that's actually a really important piece of training graduate students. Many of my graduate students want to go into industry, and so I think there's been a bit of a vacuum, to be honest. I mean, even when I was going through graduate school, and understand what that was going to be like, and maybe what the expectations would be. And so as, another hat of being a recruiter for Amgen, I've been on the other side of that, and understanding what people are looking for and how you bring in the best people. So I want to train those people, right? And I want to give them the best chance of success of getting that job and being really great at it. And so, what I think is important is to be realistic and give them a real flavor for what they're in for, and the things that they should be thinking about in year 1, not year 4 or 5, right? So really training them to think about the long game and how they can be productive and how they hone those skills appropriately, that they can be the best at whatever they choose. And if they choose to change direction, that they can also do that, right? They just need the right skills, and they need to be thinking about that. I think that's a really important aspect of what I try to do.
Yeah, I agree with that. My lab is very diverse in the sense that we have projects that are highly biochemical or structural biochemical. Some are just mice, and other is translation. And so because of that diversity, my students and my postdocs are basically exposed to the both ends of the spectrum. And I think it served well because some of them, left my lab and straight went into the industry. They got a job offer right after. They are actually after them before they complete the thesis, and so I had to slow them down. And some others ended up in academia. But I always stress, I'm a mechanistic guy, and always stress that mechanism. It's the answer to find novel pathways or molecular pathways that are responsible for the pathogenesis of certain disease, but find targets that they can be translated into translational medicine. I do have students that come to me with expertise in drug screening. They see my pedigree and they come and they want to do more drug screening. And some are interested in mice, and so they come to do the behavioral part. I accept them both. I mean, just, you can help them establish their career and fulfill their needs, their desires. At the same time, while doing drug screening, I tell them, you should also have a project in something else, and so it gives you in something else and gives you a more global view of the whole spectrum.
Great question. Thank you. So I got one last question. Based on your academic drug discovery pursuits and experiences, what are the key gaps that WARF Therapeutics could address?
All those that you're trying to solve for me right now.
We'll start on Monday.
Nice slide that you had put together, little puzzle pieces. Actually, the context is really bridging the pieces. There are things that we do very well on this campus. We've seen examples of that today. But I do think that really translational and getting things across the plane to clinical trials and such is going to take a robust effort. It's a team effort, and there's no individual. And so I think the better teams that we can build and the more resources we bring in, the better off we're going to be.
That's a great response, Jennifer. And my experience has been that drug discovery is a team sport. Right now, WARF Therapeutics is a team of one. It might grow to a few more. And so, I'm going to need to, leverage, the campus community to help move these things forward. Rely on, our advisory board and, consultants to help fill sort of that virtual intellectual, role. But at the end of the day, it's a team sport. Nobody has all of the ideas and all of the solutions. We showed 8 compounds that I moved into the clinic during my last few years at Merck, and I can say that for many of those, I have a area of that molecule that I contributed that was meaningful, but without the influence and the creative ideas from the rest of the team, there wouldn't be any molecule in the clinic, and so it's a team sport.
Actually, I have to say that I've been doing experiments, and I have projects that are the result of my conversation with WARF. Because there are things that I wasn't thinking of, because obviously I'm after mechanism. But then I realized that there are questions that can be studied and that can actually go into the mechanism and can expand the research. And so it's been a mutual, it's been a learning.
It'd be useful even to have sort of a consulting service. It's what you've even heard here is that we all have different gaps for each of the 3 programs that we've had. And having as a resource, maybe at WARF, to be able to figure out, here are the people that you need to talk to. Central clearinghouse for that, rather than people feel kind of left to figure it out themselves.
I actually loved that aspect of one of your slides, John, where, even if someone puts their program in and perhaps it doesn't get adopted into the pipeline, is that you'll still get feedback, right? You'll still get feedback, and you'll still be posed back these questions of where you might reapply, or maybe more fundamentally what you could think about to move the program in a different way. And so, I think these guys have both mentioned that.
Terrific. We have time for perhaps one last question, if there's one.
Great presentation. This is sort of a general question. We sort of know industry, academics is public and industry is private. But what about philanthropy? How does that affect what you do? And we know what Bill Gates has done for malaria. What percentage of your work could be supportive like that?
I think that one came to me. So, it's a really good question. There's no doubt that part of the WARF mission is to be able to give back financially and in other means to the campus. And so I think the majority of what we're going to be working on, therefore, is something that hopefully can take that initial investment and turn that into a revenue stream that grows over time. And so from a philanthropy perspective, I guess we're in essence meeting that goal, because we're giving back to the university, to the laboratories, and so on and so forth. Pursuing drugs for unmet needs that may not be revenue generating, that's certainly something that we can consider, but I think that's going to be a challenging area. And I think some of the work that Jennifer is doing, and I know I have a lot of colleagues that work for the Gates Foundation. I know that there's parts of Celgene and Merck and other organizations that are probably better equipped to be able to go after those particular types of diseases. Beth,
So I'm Beth, I'm at WARF. I've been working in the pharmaceutical portfolio for my 14 years at WARF. I just want to stress WARF Therapeutics is a great opportunity for researchers who have come to me in the past, and I've had to tell them I'm sorry, there's nothing I can really do to help you. Now there's somebody who can potentially help you at WARF. We can take these early stage assets and help to develop them. That's just one part of WARF though. So we're still interested in drug development technologies. We still have avenues to patent drug development technologies. We have business development people here who are looking at unmet needs, trying to find industrial partners, philanthropic organizations, whereas WARF Therapeutics may not take those technologies on. There are other things going on at WARF, I would say, to try to address these other aspects of pharmaceuticals. So feel free, reach out. We love to talk to you. If it's not WARF Therapeutics, there are other things we may be able to plug into to help develop these technologies further.
Terrific. Well, I think that concludes our presentations here this morning, and I'd like to thank our panelists for a really nice conversation. Perhaps we can give them a round of applause, please. And so over the coming days, there's a survey that's going to go out to all of the attendees. Please take some time to fill that out. We'd love to hear from you. We're trying to look forward in time to other opportunities to talk about our new programs, some perhaps different directions, but also opportunities to help continue to build a drug discovery culture here on campus. And so please take the time. We'd love to hear from you. And once again, thank you very much for your kind attention, and look forward to working with you in the near future. Thank you.